Thank you very much, and welcome everybody. So first of all, just to share my conflicts of interest, and as you can see, I haven't updated my profile photo for 20 years. But yeah, it's going to happen one day. It will happen one day. There's my disclosures. So let's focus on our panel today. We have a very distinguished panel from all over the US and Pina as well from Italy. So if I start with Raj, so Raj is based in Chicago at the moment, and as you'll know, has a keen interest in both clinical and research activities. He's working across the inflammatory domain, and he always has really cool shoes. You know, look at him, look at him. I realise when I'm up here and he's like, I'm so boring. I've shaved my head, but I've got boring shoes. But anyway, welcome Raj. And then we've got Tina as well. That's right.
Get up there.
He was really excited.
Right, yeah. And we've got Tina Porter, who's come from Boston. And Tina, like Raj, has a big academic and clinical interest. I think she focuses a little bit more in the HS space and also the IBD-related dermatoses. But of course, she can cover questions on all inflammatory dermatoses and has a lot of clinical trial experience. She's also wearing much cooler shoes than me.
It's only because I knew Raj was going to wear sneakers.
And then finally, last but not least, is Pina D'Angelo. And Pina, I found out you're a big Napoli fan, but she has been to Old Trafford, Manchester United, which is a great shame. Although I'm based at the University of Manchester, big Liverpool fan. Okay? But Pina, importantly, a mathematical and statistical background from McGill, but has over 25 years in clinical research experience, and in the last 10 years that focus has very much moved towards dermatology. So welcome all of you. It's pretty difficult this, I'm going to fall off this stage I reckon if I'm not careful. Let's move this on. So I'm going to hand over to Raj first of all. So we've got a couple of short presentations. Raj, then Tina on AD and a bit on HS, and then we'll get into the nitty gritty and Q&As. So Raj, over to you.
Thank you. Thanks so much for having all of us here to talk about what's been a very puzzling issue for many of us that are in these disease spaces that we're focusing on today. And I know I have just a little bit of time, and so what I want to do is maybe highlight a little bit about where we are with atopic dermatitis, particularly in terms of current therapies. What are some of the issues that don't allow us to compare our therapies very easily, and why that feeds into some of the problems as it relates to unpredictability in trials, and particularly in terms of placebo rates. Here are my disclosures. And I'll start off by saying that it's been actually an incredible ride over the past decade of where we've gotten in terms of our systemic and even topical therapies. And really, we have a lot of options now that we just didn't have that go beyond sort of the basics that we used to use for a long period of time. And what we sort of do with these academic exercises at conferences is really love to put out the latest network meta-analysis comparing therapies, because really that's kind of all we have to allow us to think about relative differences. And based on who funded them and based on how they're designed, someone's going to always come out ahead. And part of the problem here is that, is it actually really even appropriate to compare the therapies we have? And if not, why is that such a problem? And so if you actually break down randomized controlled trials in atopic dermatitis, their comparability is not as easy as you might think. And there's a lot of different participant-related factors, disease-related factors, and treatment-related factors to consider. And this is just a really nice infographic that highlights what some of those might be. Everything related to inclusion or exclusion criteria, the objective measurements used, how screening is designed, disease activity itself, comparator choices, statistical analysis, how you're handling data, how things are evaluated over time, what washout looks like. And this alone should highlight what hopefully you're already starting to think about in terms of why there's a lot of areas in which potentially things may go wrong and or feed into this concept of placebo rates being a bit unpredictable in current studies. And so I'll just give you one really good example, and this is actually something that probably relates quite strongly to the placebo question, and we'll talk about in a few slides. Let's talk about rescue treatment criteria for just a moment as one of those like dozen or 24 parameters I mentioned on the other slide. When you think about it, whether you're thinking about sort of the length of your screening period, short screening, long screening, whether you're thinking about your washout period, whether you're thinking about sort of inclusion criteria and disease severity, or thinking about your comparators, really the concept of topical rescue in atopic dermatitis can actually affect the response rate to placebo. Washouts can increase the need for rescue treatment. Screening can increase disease severity and rescue treatment use. Those who may not be included in an extension trial due to rescue use can influence statistical analysis. So this again also shows you that even amongst those large concepts that I mentioned, there's a lot of different places within each of them as well that can influence exactly how your data looks too. So this sort of begins to build a case that maybe we're not taking everything into account in atopic dermatitis. And not even mentioned on that slide is another puzzling issue that we and others have published on. The idea of longitudinal course in atopic dermatitis is not taken into account at all in the current way we think about disease, meaning that there are individuals that have more episodic disease, more chronic disease, have flare patterns of difference. This varies around the world. It varies with time, and there has never been a really good way to capture this too. So there are hidden elements inside the disease state that still do not have some way to be controlled in the course of current trials as well. And it probably is another factor that starts to feed into this issue that we're talking about here today too. So what trends have we actually seen in the last decade of clinical trials? There's been a few folks that have tried to at least very grossly analyze this issue. And I'll tell you that when you start to stack up, let's say, the trials that were conducted in 2014 and 2023, thinking about things that at least had an EASI score of 16 or above, on this graph, what you're noticing as you move your way from left to right is a dot that represents a number of phase 2 and phase 3 trials for many assets that we currently have on the market, some which are not on the market and didn't go forward. On the y-axis, you're seeing EASI score at baseline, and on the bottom, you're seeing month and year of the study start date. And what you're noticing is actually a very interesting pattern that as we've gone forward in time, the EASI score at baseline has actually fairly well tracked in sort of this linear slope downwards. And if you actually break this down even furthermore and take a look, this graph is looking at sort of the similar set of studies. This is sort of 23 studies, again, the same decade, baseline EASI score at least 16 or above requirement. On the x-axis, we're looking at the baseline EASI score in the placebo groups, going from 24 all the way to 35, and you're looking at percentage reduction in EASI score at week 16. And you'll note that as we tend to have lower and lower baseline scores for EASIs and placebo groups, generally speaking, there seems to be this trend of an increased percentage reduction in EASI score at week 16 as well. Finally, one more point from this particular publication that was put forward. If we think about sort of that baseline EASI score in placebo groups and then think about the percentage of patients using rescue medication by week 16, you also notice a very clear trend too over this decade that as the baseline EASI score tends to go up, rescue medication by week 16 tends to increase as well. And so, how does this all kind of fit into this larger picture? And there's been folks that have tried to take this type of data, and this was a recent publication in the last year, that really tried to go forward with, is there a way to model this concept of combination therapy, baseline disease severity, and really predict what might be some of the expected placebo outcome rates. And so this used a type of technique called longitudinal nonlinear mixed effects modeling, which took into account trial phase, concomitant therapy, and baseline disease severity, and then tried to understand a little bit about what would be sort of the ideal protocol design in terms of time as well as points. And there was a few things to take away from this. Generally, trials permitting topical corticosteroids had nearly a twofold increase in placebo EASI 75 versus monotherapy in terms of the placebo response. And then baseline disease severity was another significant driver. With every 1 point increase in baseline EASI, there was a reduction in EASI 75 placebo rate at weeks 12 and 16 by 0.96. Another interesting point as well, EASI 75 outcomes at week 12 actually captured about 94% of the predicted response at week 16. So you can see that based on this little matrix that they designed, perhaps some of the ways that you can already start to think about correcting this issue, maybe our trials are actually pushing a little too extra in time with this driving up some of this rescue use and potentially even some of this placebo rate. The baseline EASI score and perhaps even the actual concomitant therapy question are really modifiers in terms of ways that you can predict what might be happening. So another point that really doesn't totally relate to the placebo question, and this kind of comes up all the time, does baseline EASI score actually have an impact on active treatment arms as opposed to the placebo treatment arm? And there actually aren't many analyses. This was a sub-analysis in an Italian journal of some of the dupilumab data. This was a post hoc of Kronos and AD Cafe. And what they found was that generally speaking, an EASI of less than 24 or greater than 24 actually had a similar response in the treatment arm. So this seems to be more of a unique problem for placebo groups than the actual active treatment groups as well, but a very unexplored concept beyond this particular publication. So I'll stop with this slide. I sort of put in a couple of different examples of what's been happening in trials of today, just to look at exactly how unpredictable some of the responses have been. But this was actually a nice paper put forth by the IEC right around 2020 that really tried to come up with possibilities in terms of an algorithm about where you should be thinking about trying to optimize trials. And the idea here was that inclusions of patients with milder diseases seems to be driving this eligibility creep that's maybe resulting in some of this unpredictability that we've seen. This idea of concomitant use, this idea of a clean study versus real world, we feel this tug and pull all the time, is perhaps driving this too. And then there's a lot of environmental influences, like I mentioned, that we just don't capture really, really well. And so if you kind of follow this diagram here, it's very difficult to read from where you're standing. You'll see that everything from site selection in terms of centers with expertise in research and assessments by dermatologists who know atopic dermatitis, well-defined screening, very clear specification of failure, including minimal use potency and length of time used for preceding TCS and TCI, minimal possible durations defined by medication for washout, thinking about sort of the actual optimization of the intervention phase, understanding how you're handling your data from dropout versus completed studies, and thinking about higher bars for outcomes are all places that we can probably begin to start standardize across trials to maybe address this point. I'll leave it there, and looking forward to the discussion. Thank you, guys.
So I'm going to talk a little bit more about placebo response in HS clinical trials, and actually, it's funny, Raj and I like to text things to each other, and one of the things I texted at one time was, oh my gosh, your AD trials are turning into my HS trials for the placebo response rates versus efficacy. And actually, I think what you'll see is there's quite a few themes, I think, that resonate between the two disease states, even though they have nothing really in common, I think, in terms of what they look like. But we don't have as many beautiful papers like you just showed looking at placebo response yet. There's quite a few things that go into what we consider potential contributors could be, and these are things like difficulty with disease severity assessments, because this disease is very difficult to score based on the fact that it's mostly a dermal process and there's quite a bit of scarring involved. There really is not an objective measure of disease state that we look at. So the outcomes that we look at in clinical trials for HS are either patient-reported outcomes or they're physician-assessed outcomes. It's not like doing a diabetes trial where we can monitor hemoglobin A1C. We don't have any good biomarkers to really look at this disease state. And some of the inflammatory biomarkers that we may use in IBD actually do not hold true to be good biomarkers for HS. And that's been very difficult, I think, as we look at these placebo response rates. And then actually there's no standardized definitions of lesion types themselves, and this is actually kind of a fascinating thing going forward. And I've worked on dozens of protocols now, and there's many papers, and they all define the lesions slightly differently, and this is something over the last 5 to 7 years that we've really tried to standardize. And then within the disease itself, it's very heterogeneous. If you're a lumper or splitter in dermatology, we always talk about this. I think there's probably multiple disease states that have been lumped together and called HS, and that's why we get such a different look, and why when we're doing all these different sampling studies, we get a very wide number of results. There's not like one biomarker that emerges. There's not one clear pathway. And then related to this, we actually are starting to see different responses, both by disease phenotype, which we don't have, again, a standardized way to capture, but also by lesion type. So more and more when you see HS clinical trials, you'll see the data read out as what was the response for the abscesses, what was the response for the inflammatory nodules, what was the response for the draining tunnels, what was the response for the combinations of these lesions. And some of this is important because there have been drugs actually that had very good responses for draining tunnels, but had very little impact on the abscess and nodule counts. And so when we're looking at measures like HiSCR, where you're only looking at the % change in the abscess and nodules and not really looking at the changes in the draining tunnels, these drugs don't meet statistical significance. And the question comes up, is the drug not good? Is the outcome measure not good? Or is it something in between? And then sort of like what Raj was talking about, we have different trial sizes, lengths, site selection. There's actually a lot of geographic variability, I think, in this disease. And one example of this is that in Asia, we tend to see more males than females. And in the United States, we tend to see many more females than males who have the disease, and we've never actually randomized by gender for any clinical trial that I'm aware of. But actually what we're starting to see is that some of the trials where we see differences in these phase 3 studies, one of the main features that we see is that there's gender imbalance, and we've never really delved deeper into this. And then the disease itself has a lot of underlying disease variability, and even what we're looking at now for current therapies are nowhere near where we are for atopic dermatitis or psoriasis. And then, Raj mentioned this a lot, there is rescue therapy and this idea of the concomitant meds and the washout that can affect these placebo rates as well. So Alexa Kimball and I got very obsessed with looking at placebo response, probably because everyone kept telling us the outcome measures don't work at all. And so we went through, we did a bunch of post-hoc analyses actually with the Pioneer data to go back and look at placebo responders. We were looking at hematologic abnormalities, and we looked at CRP as well. And one of the things we were trying to ask here is, essentially, like, is placebo response in HS just noise, or is there actually a real biologic improvement? Meaning, do these placebo responders actually have a response without receiving any drug? And actually what we found is that some probably do. So there are patients in the placebo group that are truly improving even though they're not on any therapy. And actually there was an article in JAMA Dermatology that just came out a couple months ago talking about patients who disease just goes into remission in clinical offices versus hospital settings, and that we've greatly overestimated the number of patients whose disease remains severe, because they showed that more than half the patients over 10 years in this Danish cohort just didn't have HS anymore, even though they were very severe. So the other things that we've seen too are that a lot of these patients have hematologic abnormalities, and even in some of the adalimumab trials, patients who had hematologic abnormalities at baseline who were in the placebo group had improvements too, suggesting that they probably also were having a clinical improvement with their disease state, because the things that we see like leukocytosis, anemia, were all resolving in the placebo group as well. And then we also were just looking at, can baseline CRP help predict who responds to adalimumab? And essentially, if patients who have a higher CRP burden at baseline do seem to have better response to adalimumab than those who do not, regardless of what their disease severity is. The problem is that CRP doesn't actually reflect baseline disease severity itself. So patients could be very severe and have no abnormalities in CRP, or relatively moderate and have very high abnormalities in CRP. So this is what we're really looking at now when we think about trial design for HS. We have minimum lesion counts for entry, and you've actually probably seen this creeping up over the years. The Pioneer study started at 4, now we're at 5, and there's even some phase 2 studies now that have moved up for AN count to 6. And this is really, part of it was John Frew looked again at the post-hoc analysis for Pioneer data, and he said if you used an AN count of 7, the placebo rate would drop significantly. The problem is in clinical practice, when we think of a moderate to severe population, an AN count of 7 is probably really skewing to a much more severe population, or it's skewing to a population that's getting mostly abscesses and nodules and has few draining tunnels, which is a different disease phenotype. We haven't looked yet at combining something like ANs and DT counts as a baseline criteria, but it's something that probably could be considered. And then one of the things we're also recommending is really tracking severity of the enrolled patients that you're having at set time points during your enrollment period, so not after the entire trial has enrolled, because what we see is based on geographic distribution of the sites that start first. Americans usually have much more severe disease than some of the European cohorts that we see. There's more Hurley 3 patients in Hurley 2, and so if you're doing a trial in just certain areas and almost all the patients come from one area, you might end up with patients who are much more severe or much less severe. And we've kind of gotten to a point where we're really looking at trying to get patients at an AN count average for the whole trial around 12, with a draining tunnel count somewhere in the 2 to 3 range, to really be able to see differences in response for the moderate to severe population. There's a lot of talk about photos, and I'll go over that in a separate slide. And then also looking, I think, at the review and documentation of rescue criteria. It's very complicated in HS. Almost every protocol says lesions that are intervened upon should be counted as permanently present, but none of the data capture systems are really set up to capture this information, and it requires a lot of training for investigators, both to actually document this, but actually to help them understand that the key thing is that you cannot rescue a patient the day one that they start in the trial, and that actually happened in quite a few trials in the past, where patients would go through these long washout periods and then they get a course of antibiotics on day 2 because they were suffering so much, and those patients in many statistical analyses are now considered non-responders. But at 16 weeks, that 7 days of antibiotics has no effect on their actual response to the therapy, but they're now in this category where they're a non-responder, and it's very difficult to understand how to interpret that data. And then a lot, I think, is also looking at randomization schemes. What criteria should we use to really try to balance the groups? And then one of the things I think that needs to happen in HS more than any other studies is sites have to be audited quite early. And there's a lot of things that go into developing these data capture systems to really look for lesion variability that is probably more rater related than disease severity related, where you see wide fluctuations in counts that just really shouldn't occur. And then flagging these sites to ensure they're not enrolling the majority of your population or providing retraining. What we do see is there is quite a difference in response by severity. There's this idea of a follicular or nodular patient population where they have very high AN counts but low DT counts. These patients are more likely to be female, have more disease variability. They're actually more responsive to medications, but the placebo rates in that group can be much higher. Whereas inflammatory phenotypes, which are a little bit more difficult to classify, are generally patients with more long-standing severe disease who are less treatment responsive. And these patients tend to have lower placebo rates but are much harder to treat, so you really have to figure out, like, the more severe you skew your trial, the less likely they'll probably respond to therapy in a short 12 to 16 week period, but the more likely their placebo response will be lower. If we're out of time, just let me know. And then I just wanted to show this one thing too. This kind of reinforces this, but in the Sunshine studies for secukinumab, they actually did a machine learning analysis afterwards, and what they found is there were different clusters of patients, and it was once they separated out these by cluster that they actually started to see dose responses between the two different dose frequencies, and it was only in the most severe patients that the Q2 week dosing actually started to show that it was needed. And this, I think, actually reflects clinical practice, but in a large trial like this, depending on where your patients end up sitting, you can have these patients masked by the other disease severity population. And then the last thing, we made this paper called HS Wobble, which no clinician really cares about, but all pharmaceutical companies do. But essentially what we started to see is that there were, if you cut off a trial at week 12 instead of week 16, you might have met statistical significance because the patients did much better at week 12 and then their efficacy just fell off at week 16. And we called this the HS Wobble, and we went back and looked at all the different studies that had been published to date, and essentially what we found is that patients who are less severe, more likely to be female, those are the ones where we saw this wobble and the placebo rates and the efficacy responses, and probably it's a reflection of underlying disease severity as well. The problem, I think, when we're looking at 12 versus 16 weeks, at least in the United States, is sometimes the FDA has requirements for the length of time that they want the patients to be on drug, and so you can't actually choose a week 12 endpoint because of some of the safety considerations. And then the last slide is just looking at technology here. I think people really want to believe that you could do what we do in alopecia areata or atopic dermatitis, where they take a photo, you can have a central reader, they'll tell you if the patient qualifies, and then everything is great, right? But the problem with HS, and we've studied this at our own site, is we track patients every month. We took photos of them in a very meticulous manner, the way that's recommended by Canfield and has been used across all the trials. And my fellow rated all the patients in person, and I looked at all the photographs, and I look at thousands of photographs and thousands of patients. There's no way I could ever assess the patient's disease severity looking at these photographs. Some patients who have more nodules are easier, but once the patients have darker skin pigmentation and the heavier they are, the larger body mass index, all the lesions become obscured, and you really need this element of palpation. I think there are ways to apply machine learning algorithms to photos to actually phenotype patients better. That can be done. Antonio Martorell did it with the AI group in Spain, and it was pretty fascinating to be able to see. And we could also consider things like ultrasound or other imaging modalities to fill this gap for dermal lesions, but it's, again, very difficult and operator dependent, and I think all of this requires a lot of time and training, and I'm not sure that this is where we want to spend all our time in a trial trying to train someone to take a photograph or do an ultrasound versus spending the time really trying to help them assess the disease better. Okay, and this is my last thing. Probably more questions than answers, so please feel free to ask later.
Tina and Raj, so we're going to go into a panel discussion where we've got a few questions set up that I'll ask the panel. I am hoping to take some questions from the audience as well. So we'll try and keep our answers fairly succinct through this, but obviously, because we have covered some very important stuff within these discussions. So question 1, do you see value in clinical trials for mandatory photos? Well, I think we've already heard from Tina in the HS domain where that may be problematic. Screening to try and ensure patients are severe enough. And I guess this does relate to HS. You've kind of given an opinion on this already.
Oh, yeah. I do think that you can do early staging in photographs quite easily. The other caveat is that most patients with HS have them in areas that are very sensitive and they do not like to be photographed. And so unless you're doing essentially total body photography where you're looking at every single area that has HS, it can be a little hard. If you miss one area, it'll affect your interpretation of both their disease severity and their improvement.
Raj, do you want to come in? I mean, maybe broaden this discussion across other disease states where it may work a little bit better.
Yeah, so we can talk about photography and atopic dermatitis, and I'm of two minds about this. And so Tina is eternally sunny and positive about atopic dermatitis photography, but sometimes it's not so entirely straightforward, right? So alopecia areata, vitiligo, they have a lot of things going for them where central read can really make this quite simple. For atopic dermatitis, I think that photography does have the power to potentially help confirm diagnosis in a lot of cases and maybe even ballpark severity. The problem is that it can be actually quite difficult based on the quality of the photographs, the secondary aspects of the lesion you're looking at, erythema, particularly across darker skin tones, as well as some more subtle secondary changes, especially when you're starting to look for lichenification and other things that do make a really good grading of severity tough. But it does feel like if at least the necessity for photographs is there, there's a little pressure on the individual that's considering enrolling the patient to really consider somebody that's going to have a relative BSA, IgA, and other parameters that fit what they're looking for. I think that we hear oftentimes for central read and atopic dermatitis that people are able to catch things that aren't AD. The problem is AD is so heterogeneous that even that's a little tricky too.
So one thing I've been doing recently is in palmoplantar pustulosis with photo adjudication, and I think one of the big things there where it has been helpful is separating dyshydrotic eczema, which can be mislabeled. So it is useful, but even then the photos coming through, the hyperkeratosis you get on the skin can actually mask the pustules, and it's very, very difficult on some photos to always clear to know how many pustules there are. So photos are good. I think they absolutely have a role, but I think you have to think very hard about how you employ them, how it affects recruitment, how it affects the screening process, and the photo adjudication has to be extremely rapid not to hold the trial up.
Yeah.
So I'll move on to question 2. In your opinion, what is the best practice to ensure a proper baseline severity of patients without inducing bias in patient selection? So I think all three of you are going to comment on this. I'll perhaps come to Raj first, then Pina, and then back to you, Tina.
Okay, so this is also a challenge as well, given just what I showed you in terms of the trends of overall severity and some of the issues that we've seen and many of you heard about and heard from all of us talk about these individuals that are on the low end of moderate disease that are really sort of on the cusp of making it into a trial itself, and a lot of inflation that comes with some of these scores in a group of patients that oftentimes are a bit variable. Now, this is a little bit of a challenge given the fact that there's a volume of patients needed, and oftentimes the sites that you want for people that you think are going to do very good scoring have limitations in terms of how much they can actually bring into the trial. The photos can at least help here, particularly in terms of some education of IgA. If you're looking at a couple different body sites to confirm if the ballpark severity assessment is reasonable. I think, again, the training part of it, and I know most people get annoyed by it, being extraordinarily aggressive about training and retraining can make a big difference too. Site selection, you're just going to have to understand the time frame may not meet what you're looking for, but that probably will allow you for at least a better severity angle. And then now what we're seeing is actually capping of severity too, and potentially trying to actually have a limitation in the low end of moderate in a range that does not sort of kill your power as well. That's all nice, and I'm not sure if that'll even solve the problem, but I think that level of aggressiveness is probably going to be needed.
Thank you, Raj. Pina, did you want to come in on this one?
Yes, thanks. So what I would say is it's really important to detail this in the protocol. So sometimes what we see is that if it isn't detailed in the protocol and that we're not, we're seeing averages or medians of baseline severity that is not what it wants to be, we start introducing some bias by doing things that the agency may not be quite on board with. The other thing also is to stratify, so it's not going to control it, but at least you'll have a good balance in your treatment between the lesser severe and more severe patients.
Thank you very much. And Tina, applying this maybe to
Yeah, I mean, I think I touched on this already. I think the most important thing is establishing caps, not to ensure that you have one type of patient population over another. It's actually to have a wide range of patient populations. And that's where I think you have to have a little more experience, I think, to know where you're expecting to get certain patients from. Like I said, we'll see different trends based on geography, and so we'll know we need this many sites, and these sites contribute this many number of patients to try to get enough these essentially mild or moderates to the more severes.
Okay, thank you. So the third question, in atopic dermatitis trials, baseline severity is known to be correlated to placebo response. Raj, you actually did articulate this. What is the impact of an incorrect IgA evaluation at baseline on placebo response? So,
I think we kind of covered that. I think we covered that reasonably well, but just to re-emphasize, not that one wants to accuse individuals of investigatorial misconduct, but generally speaking, if your IgAs are not truly reflective of what your patient is, that does unfortunately have the ability to introduce a lot of this variability that does seem to play a role in some of the unpredictability, particularly in phase 2 that we've seen over the last few years.
Pina, I think you may have some additional points as well on this.
Yes, please. Can I ask, how many of us are statisticians in the room?
None.
We're all idiots.
Oh my goodness. I'm in a room full of clinicians. I'm in a lot of trouble.
I'm not a statistician at all. Sorry.
So just a little bit of background, and I'm mindful of time. We've taken placebo response really seriously at Indero. We have done an extensive amount of R&D work. A little bit of statistics with respect to this. So we actually did a post-evaluation of photos. So we had the equivalence of an independent medical person. Many of these were done actually by Dr. Rosalind, who used the photographs and did his own assessment of the IgA. And we also do this through our independent... Sorry?
More than 6,000.
Yes, more than 6,000 photographs. And we also have our own independent medical monitors in our ongoing trials who perform this as well. So if we imagine taking a subset of these patients where the IgA that was assessed by an independent person, Dr. Rosalind or otherwise, versus the IgA that was scored by the clinician during the trial. And if we just take the subset of patients where it was felt that that independent assessment did not meet the protocol requirement. So for example, a PI may have scored an IgA of 3, the inclusion criteria was moderate severe, and the independent, Dr. Rosalind or someone else's opinion might have been that the IgA was 2. We look at these patients. If we compare the placebo response with respect to an EASI-75 or a clinical response on the IgA, it's actually 3 times higher. So this subset of patient is really, really problematic. So I know it was, according to also our presenters, we know this, but the magnitude is huge.
Okay, thank you. That's very clear. So placebo rates vary by geography, and if they do, why is this the case, and how can we address it?
Pina.
I was going to say statistically, what is your crumble crunching show?
You guys are terrible. I'm deciding who wants to first. No, it's fine. Pina, please go.
Take it out of your hands.
Yeah, go on.
Yeah.
Right. So what I'll say is that the data that we have in-house, we have crunched these numbers. I don't feel confident that that sample size is big enough to make a statement to this. What we're seeing right now is there is none, and we're looking in North America and Europe primarily. The literature doesn't suggest anything. What a recent publication, actually quite interesting, did show that the only thing they were able to find was a correlation in general placebo trials, so not in the indication of dermatology or the therapeutic area of dermatology, but there is a negative correlation with respect to socioeconomic factors. So a high, or I should say a low GNP will have a high placebo rate, and that's usually owing to the fact that a patient may live in a place where healthcare is absolutely not accessible. Just the fact that they are on a study, they're thrilled, someone is taking care of them, and so we see a placebo rate going out. So right now, that's kind of the only statement I'm comfortable with.
Interesting.
Okay, thank you.
Yeah.
Raj?
So, I'll add a couple of thoughts. Some of the trends we do tend to see, at least in the atopic dermatitis world, there's definitely different handling in terms of what people are sort of calling the disease state itself from place to place, particularly in North America versus certain places outside of North America and certain parts of Europe. And so that does lead to a bit of confounding. Particularly, I know we're talking about atopic dermatitis. We see this issue with prurigo nodularis even more than atopic dermatitis. Another potential issue and a real issue is sort of the use of concurrent therapy and potential rescue therapy, given what sort of the actual protocol is going to say. There are certain countries that are just more aggressive in terms of what they rescue with, how they rescue, how quickly they're going to, and how much hardcore topical corticosteroid use there is as well. And we showed in that data that I cited that indeed that has a bit of an effect too. And then finally, I showed that graph of just theoretical courses of disease. We know atopic dermatitis is a disease of intrinsic and extrinsic, and the exposome changes so much of not only the immunologic heterogeneity of the disease, but even sort of the clinical heterogeneity. And there's never been a really great way to capture it, but we know that there's differences based on humidity, sunlight, diet, water, pollution. A lot of that's tied into social and structural determinants of health in terms of where people live. And so this really creates a very complex and difficult to control because we don't really measure those factors in sociodemographics. We're very obsessed with race, age, sex, etc. But none of those really sort of captured those points.
Yeah, and actually, building on that, there are some studies now in HS looking at healthcare utilization based on times of the year, essentially, because if you talk to patients, they'll say their disease tends to flare when it gets hot in the summertime. And there are certain temperatures or climates where we have a lot more temperature variability. And in Boston, for example, the number of calls we get for flares in the summertime is exponentially larger than what we get in cold temperatures or the winter or when the seasons are changing. It's hard to say if that would truly affect some of the responses that we see in a clinical trial because nobody's on the exact same time points. But the other thing I think that sort of happens with geography is, like I said, HS is really a bunch of diseases that are probably grouped together. And for example, in Asia, it's mostly men and they get a very different looking disease state. And originally when they were looking at the potential genetic contributions to HS, the thing that came up a lot was these gamma secretase mutations in patients who have very severe disease. And actually, if you look at who has this in the population, it's probably more males and really a lot more concentrated in Asia. And those patients also are thought to be more of this like autoinflammatory syndromic category where IL-1 has always been implicated in things like POSH syndrome or PAH types of things. And so you may actually see differing responses in terms of efficacy, not necessarily the placebo rates by the geographic regions. And it can vary from drug to drug because of the mechanism.
So I think with that, we will move towards closing the event. So first of all, I just want to thank the sponsor, Indero, for putting this together. I think it's been a very interesting discussion. And that, of course, has been facilitated by our excellent panelists. So Tina, Raj and Pina, thank you very much. Clearly, I think this is something that goes on each year at AAD, and I'm sure Indero would be keen to hear feedback about what you think about this and other ideas that may be useful for future years. So please feel free to do that. I was thinking that it's a join the community rather than I thought it was a feedback QR code, but maybe give feedback directly to the team.
They don't want you
join the community if you wish. All right, thank you very much. Have a great day.